Good morning, dear students. So in last class, we were discussing about the meristematic tissue. So today we are going to start with the next topic of the same chapter, that is permanent tissue. So in last class, we discussed that what is meristematic tissue, what is apical and what is lateral and intercalary. Now today we are going to start with next, that is permanent. So let's start. Permanent tissues. Now what is this permanent tissues? Permanent tissues may be defined as group or collection of living or dead cells formed by meristematic tissue and have lost their ability to divide and have permanently placed at fixed positions in the plant body. Permanent tissues are classified into two groups, that is simple permanent tissue and complex permanent tissue. So in simple term, we can say that permanent tissue is originated from meristematic tissue only and becomes permanent at fixed positions in the plant body. They are made up of mature cells that have undergone growth and differentiation. Permanent cells possess definite shape, size, and functions. They have lost their power of division. They may be living or dead. The living permanent cells are large and usually possess thin walls enclosing vacuolated cytoplasm, and the dead permanent cells are usually thick walled without cytoplasm. So we will be starting with the first one, that is simple permanent tissue. Before heading, we will see what is the difference between meristematic tissue and permanent tissue. So what are the some differences? First point, if we see, its component cells are small, spherical or polygonal and undifferentiated. But in permanent tissue, its component cells are large, differentiated with different shapes. Next, cytoplasm is dense, vacuoles are nearly absent, but in case of permanent tissue, large central vacuole occurs in living permanent cells. In meristematic tissue, intercellular spaces are absent. In permanent tissue, intercellular spaces are often present. In meristematic tissue, cell wall of its cells is thin and elastic, but in permanent tissue, cell wall of its cell is thin or thick. Nucleus of each cell of this tissue is large and prominent, but in permanent tissue, nucleus of each cell of this tissue is less conspicuous. Next, in meristematic tissue, its cells grow and divide regularly, but in permanent tissue, its cells do not normally divide. In meristematic tissue, it is simple tissue. In permanent tissue, it can be simple, complex, or specialized. In meristematic tissue, its cells are metabolically active. In permanent tissue, metabolic rate of cells of this tissue is slow. In meristematic tissue, cells, organelles of its cells are simple. In permanent tissue, cell organelles of its cells are very or are well developed. In meristematic tissue, cells of tissues do not contain crystals and other inclusions. But in case of permanent tissue, cells of this tissue possess crystals and other inclusions. In meristematic tissue, its cells are living, but in permanent tissue, its cells may be living or dead. Next, in meristematic tissue, it provides growth to the plant, but in permanent tissue, it provides protection, support, conduction, photosynthesis, storage, etc. Here, so this is the basic difference between meristematic and permanent tissue. Now, next we will start with one activity to understand that the structure and function of various types of permanent plant tissues. You have to study anatomy of the plant organ, such as root, stem, leaves, flowers, etc. For studying the plant anatomy, you have to cut this transverse section or slice of internode of young and old stem. Stain them with safranin and stain mount the stain section in glycerin and study the slide. So in this way, we have to see the cross section. Now, what is the benefit of by doing this? Study of gross internal structure of an organ as observed in a section is called anatomy. So in other words, anatomy is the study of the way in which tissues and organs are arranged in organisms. So one activity is given in order to see what are the tissues which is arranged. So what we have to do, section cutting, it involves the following steps. How we have to perform this? Select a piece of stem, monocot or dicot, any, having 1 2 diameter, and keep it in water in a watch glass. Second point, cut a cylindrical piece of potato, carrot or radish and make a vertical slit in it. This is known as pit, and keep delicate cutting material erect and gives a firm hold. Next, insert the material for section cutting into slit made in pit. Keep the pit holding the material in your left hand. You can see in the diagram. Hold the razor in your right hand with blade facing you and handle at right angle to it. Start cutting transverse section with uniform but fast motion from outside facing you. Collect the section cut of the edge of the blade with the help of a fine drawing painting brush and put them in a clean water in a watch glass to avoid drying. Select a thin slice of uniform thickness and stain it with safranin. Mount the stained section on a clean slide in glycerine, cover it with a cover slip and study under the microscope. So that's how we go through the anatomy in terms of monocot or dicot stem. That is the transverse section cutting of the stem. And this, by doing this, we can observe different types of tissues. What are the things we are observing? You can see epidermis, then ground tissue, then vascular bundles. Now next is simple permanent tissue. What is the simple permanent tissue? These tissues are composed of cells which are structurally and functionally similar. Thus, these tissues are made of one type of cells. That's why it is known as simple permanent tissue. They are further grouped in under three categories. That means further divided into three, that is parenchyma, collenchyma, and sclerenchyma. So simple permanent tissues are those which consist of similar kind of cells and they perform similar function. They are considered as simple permanent tissue. So first is parenchyma. Now what is parenchyma? Parenchyma, if we divide this word, then para means beside, in chyma means infilling. Or in another term, if we say that para means beside, in kin means to pour. Clear? Then enchyma means to pour. So parenchyma forms the bulk of the plant body. Parenchyma cells are living and possess the power of division. The cells are rounded or isodiametric, that is equally expanded on all the sides. The parenchymatous cells are oval, round, polygonal or elongated in shape. The cell wall is thin and encloses a dense cytoplasm which contain a small nucleus and surround a large central vacuole. In other words, parenchyma cells have living protoplasm. Intercellular spaces are abundant. That is, parenchyma has loosely packed cells. Now, where it is present occurrence, the parenchyma is widely distributed in plant bodies such as stem, roots, leaves, flowers, and fruits. Thus, the parenchyma tissue is found in the soft part of the plant, such as cortex of roots, ground tissues in stems, and mesophyll of leaves. It is also distributed in pith, medullary ray, and packing tissue in xylem and phloem. You can see in the diagram also, children, that is thin peripheral layer of cytoplasm. Then it consists of nucleus, large central vacuole, Then spaces also you can see, that is intercellular space, and then cells primary, that is cell wall and rounded cell. So that is the simple parenchyma tissue. Now next is function. What is the functions of parenchyma? Basically, parenchyma serves as packing tissue, that is to fill the spaces between other tissues and maintain the shape and firmness of the plant due to its turgid cells. Second, due to turgidity property, parenchyma acts as a primary support to the stem of herbaceous plants. Next, its main function is to assimilate and storage of reserve food material. It also transport materials occurs through the cells or cell walls of parenchyma cells. Clear? That means for the process of transportation as well. Next, parenchyma cells are metabolically active. Their intercellular air spaces allow gaseous exchange. Next, they also store waste products of plants such as tannin, gum, then crystals, resins, or inorganic waste, etc. Some parenchymatous cells contain chloroplast and perform photosynthesis. Such a tissue is called chlorenchyma. In hydrophytes, large air cavity is present in parenchyma to give buoyancy to the plants. Such a type of parenchyma is called aerenchyma. Now, what is this hydrophytes, children? Hydrophytes are aquatic plants such as water hyacinth, that is hydrilla. So they are aquatic plant, and basically they are having large air cavities, and that is present in parenchyma to give buoyancy to the plants. Yeah, so such kind of parenchyma is called aerenchyma. In xerophytes, that is arid plant, example succulents, plants having fleshy parts, parenchyma acts as a water storage tissue. So these are the functions of parenchyma tissue. Next is collenchyma. Basically colla means glue. Okay, colla means glue, and enchyma means an infusion. So collenchyma tissue also consists of living cells. It shows many features of parenchyma, but is characterized by the deposition of extracellulose at the corner of the cells. In collenchyma, intercellular spaces are generally absent. Collenchyma are elongated in shape. They often contain a few chloroplasts. Now, where it is generally present, the cells of collenchyma are located below the epidermis, that is hypodermis, of dicotyledons, stem, and petiole, that is leaf stalk, that is in outer region of the cortex. These cells also occur in the midribs of dicot leaves. Collenchyma is absent in monocot stems, roots, and leaves. Now, what is the function of this collenchyma? Collenchyma is a mechanical tissue in young dicotyledonous stems and provides mechanical support and elasticity. Thus, collenchyma provides tensile strength with flexibility to those organs in which it is found. It allows easy bending in various parts of a plant without actually breaking it. When cells of collenchyma contain some chloroplast, they manufacture sugar and starch. Yeah, so that is about collenchyma. Now next we have to see, that is this is the diagram of collenchyma. So in this you can see first one, A is representing basically the transverse section of cells, B transverse section cells showing deposition of cellulose at the corners. Yeah. Now next if we see, that is difference between parenchyma and collenchyma. Now what is parenchyma, children? The tissue consists of thin walled living cells, that is known as parenchyma. Collenchyma The tissue consists of cells having localized thickening in their cell walls. Second, it is distributed in almost all the parts of the plant body. And in collenchyma, it occurs mostly in aerial parts of the plants and is restricted to other layers. Parenchyma, the living cells of parenchyma assimilate and store food. They also store waste product. But in collenchyma is the chief mechanical tissue in parts of a young plant, particularly in the young dicotyledons, that is stem. Clear? So today we have covered till parenchyma and collenchyma. In next class, we'll be starting with sclerenchyma. Thanks. Happy learning.